Hello everyone. Now I will go through practical 5, reproduction in plants. If you have not done this practical, please pause this video and complete this practical before you proceed further. Now the aim and objective of this experiment is to allow students to observe the reproductive parts of a flower and emphasizes skills in the following, which is manipulation, measurement and observation, presentation of data, observation and analysis, and conclusions and evaluation. Now you're provided with insects pollinated flowers. Alright, now so those flowers given to you are lily, heliconia, daisy, and hibiscus. Now you're instructed to cut a longitudinal section of M1 flower, which is the lily, to review its reproductive parts. Okay, now so you need to make a drawing of the exposed longitudinal section. Take note of the section needed. Okay, now another very important instructions for this part of the practical is your drawing. Now so you need to make sure that you label these 6 parts and name them in order to get the 6 marks. Okay, now in your drawing, ensure your drawing has neat drawing, clear connected or continuous lines, horizontally straight label lines, and then also at least your drawing take up 70% of the spaces given. And lastly, the line of measurement indicated on your drawing. Now this is an example how your drawing should be. Alright, so I will start with the title. The title should be longitudinal section of a lily flower. Alright, so this is a complete title. That means you give section, you also give the name of the flower. Okay, now next, if you look through very carefully, all the labels start from petal, stigma, anther, style, stamen, filament, ovary, ovule, sepals, and receptacles, as well as pedicel. Alright, so each of these must be correctly, accurately labeled, as well as spelled correctly in order to get the half a mark for each of these labels. Now, also take note that your drawing were worth 1 mark. Okay, now so the drawing need to be neat. The lines need to be dark and clear. Okay, as well as the whole drawing take up at least 70% of the space. Okay, now next look at the magnification. Magnification is drawing size. That means which is your drawing, this particular drawing that is shown here, divided by the actual size, okay, of the flower. Alright, now so from there you can calculate the magnification, which is maybe 1, 2, 3, or maybe up to 5 times. Now next, identify 2 visible features that would indicate that this flower is pollinated by insects. In other words, how do you know that this flower is insect pollinated? Alright, now so you can actually give any 2 of the features given here. You can talk about sticky stigma. The stigma is sticky. Secondly, you can talk about the size. The flower is large, conspicuous, colorful petals. Okay, now and then also talk about the nectar guides on the petals. You can actually see the nectar guides, as well as the stamen and stigma do not protrude out of the flower. So in another words, I mean the stamens and stigma are relatively short. Okay, so they stay within the flower. Okay, so they do not hang out of the flower. Okay, now so any 2 of these features will be enough, okay, to answer this question, to show that it is insect pollinated. Alright, now third question, identify 2 visible differences between M1. M1, if you take note, it is lily and another flower. Now, so this question depends on which flower you are comparing with. So if let's say you compare with heliconia, you compare with daisy or hibiscus, you need to name which flowers you compare with. Okay, now, so if you compare lily with another flower, for example, daisy, the flower is much smaller. So you can actually talk about the large, showy petals of the lily, whereas the daisy have a much smaller petals. Okay, or some people, they compare the color. The daisy is yellow in color, small petals, whereas the lily, they has a much bigger, okay, white petals or pink petals, depends on what type of petals available during the practical session. Okay, now, so besides petals, you can compare anthers, you can compare stamens, you can compare the carpels. Okay, now the carpel. So these are some of the things that you can compare. Now next, the structure identified in 1F contain reducing sugar that allows the pollen grains to germinate. Okay, now, so this is a clue that what the question is driving at. So describe briefly how you would safely conduct a test to prove that this structure contains reducing sugar. All right, now, so according to apparatus given during practicals, and then also the water bath is being prepared. So you actually know that this is actually reducing sugar test, which you all have learned in year 1 food test. Okay, now, so in your description, this is a 4-mark question. So in your description, you have to write down some of the details, okay, how you actually do the experiment, how you extract, okay, the reducing sugar from the flour. Okay, now, so there are quite a few parts that you can get the reducing sugar, either from the stigma, but you know that the stigma is relatively small, so it is not really that much of sugar that you can obtain from there. But there's also another part which is at the base of the carpel or petals. Okay, now, so where you can get the reducing sugar, or maybe you can get the sucrose. All right, now, so if you are getting from all these places, you can actually put 2 or 3 drops of distilled water to wash off some of these sugars, or 2 or 3 drops of this water at the base of the carpel or petals, okay, to obtain this sugar. Now, after that, you add 1 to 2 cm cube of the, depends on the amount, you add the same volume, okay, same volume of the Benedict solution. And after that, you heat, okay, in a boiling water bath at 90 degrees, okay, for about 3 to 5 minutes. All right, now, so just take note that during experiment, when you do all this, you have to be careful that you are dealing with hot objects. Okay, now, so you have to use test tube holder or tongs, okay, in order to bring or put this Eppendorf tube or test tubes into the water bath. Okay, now, of course, you also need to carry out a control setup. So you use a 2 cm distilled water plus 2 cm cube of Benedict solution. Okay, now, and then last part, very important, many students usually miss out this point. You have to give the positive color as well as a negative color. Now, so the expected positive color will be brick red if there is a lot of reducing sugar present. But if, let's say, there is a small amount of reducing sugar present, it will show greenish yellow, or greenish yellow in color, or greenish in color, okay, after putting in the water bath. Now, the negative color is that the solution remains blue. Okay, so that means no change even after 5 minutes in the water bath. Next, remove the part you identified in 1F and conduct the food test described, okay, in 4. Okay, now, so the solution will turn from blue, the copper blue, to give a dirty green orange precipitate. Now, actually, if you read into the details, this is a reduction reaction in which the copper turns from copper 2 to copper 1. All right, so it turns from blue to green or orange, mainly is because it convert from copper 2 to copper 1. Okay. Now next, draw one pollen grain in the left box. Okay, save the right box for the drawing of pollen grains from another flower. Okay, now so if you follow this, the left box actually is the pollen grain of the lily flower. Yeah, so this is how the lily pollen grain of the lily flower looks like. It is quite spiky, whereas the pollen grains of the grass flower is relatively smooth. All right, now so just make sure you put in the magnification. If you can, show your calculation, how you get the magnification. I don't think it is 10 times. It may be 40 or maybe up to 100 to 400 times. So, okay, this one depends on the practical, the microscope, and the degree of the magnification that you used. Okay, next, label the two enlarged scan pictures of a fresh amount with the following structures. Now, so it's good for this kind of question, they already given you what are the things to label. Okay, now, so of course, you need to identify correctly where are the which structures is which label. Okay, now so on the left side, you're given the couple or pistil of a grass flower. Okay, now so this is a grass flower. The grass flower generally they are wind pollinated, so therefore you know that the stigma is feathery, and then that is the style. Okay, and then the ovary is the swollen structure at the base. All right, so stigma, style, ovary, this make up the couple or pistil. On the right hand side, it is the stamen. Okay, now so don't mistaken. Actually, the filament is very fine. You can't really see it very clearly, but it is just a very thin white hair appeared down here in the pictures. Okay, now so the actual anthers is actually the two sausage-like structures or two tubular structure. All right, now so the white tubular structures are the anthers. Okay, and then the fine hair-like thing that is sticking to the anthers are the filaments. All right. Okay, now so this is actually highly enlarged. The actual flowers, the grass flower is not this big. This one enlarged at least 5 to 10 times. The actual flowers, one of the days I'll bring to the class and then to show you, it is actually a very tiny flower. Okay, all right. Now so even tinier than the your finger, your tiny fingernail, right? So the size of your tiny fingernail. All right, so it's just that small. All right, now the last question. Now compare the characteristics of an insect pollinated flower and a wind pollinated flower. So these are the features that you can compare. Okay, you can compare the petals, the nectar, whether is it centered or not, and then the stigma. The stigma, how does it looks? Whether is it feathery or is it compact, small? Is it protrude out of the flower or is it within the flower itself? All right, you can also compare the stamens. You can compare the pollen type, the texture of the pollen, and then also as well as the nectar. But I think the nectar guide has been done for you, okay, in this question. All right, now so this is a table of comparison. All right, you can check through your notes. Okay, you should have quite a similar table in your notes or maybe in your textbook.